Hey folks, and welcome to another edition of Zog Science. Today we're going to be talking about some of the extensions to Mendelian genetics and basically why Mendel did not get it 100% correct when he first proposed his rules. So the truth is a little bit more complicated than what Mendel told us. So it turns out that Mendel got really, really lucky, and how he got really, really lucky was that the traits that he chose to look at, the characters that he chose to look at, all followed those basic principles of dominant, recessive, independent assortment, and segregation. And that's not actually what happens for the majority of traits. And the reason why is that only a few of the traits are actually only dominant and recessive. Many genes on one chromosome, so they're not always independently assorted. He didn't know about chromosomes back in the day, so we'll give him a pass on that one. We can have single characters being impacted by multiple genes. Just some other wild and crazy stuff that we'll get to kind of talk about, as well as the environment. So let's talk about the different types of dominance that we can see. We've got our regular complete dominance, where you've got a dominant and a recessive, and those are the only two phenotypes that you're going to see. However, we can also have incomplete dominance and codominance. In incomplete dominance, we're going to have the heterozygous individual showing a blend of both homozygous traits. So this is an example of that right here. We've got a red flower being crossed with a white flower. When the gametes come together, they're going to produce a pink flower. So that is a blend of the two of them, right? Red and white, put it together, and you get pink. Now, if we're going to take the two pink flowers and cross them with each other, we're going to get back out the red and the white in the homozygous individuals, but the heterozygous individuals are going to remain pink, okay? And basically what's going on there is that the pigments for the red and the white, they're both being expressed, but they're being expressed on a very small molecular level, and so therefore we perceive it as being pink. We also have codominance. This is where a heterozygous individual shows both homozygous traits. So over here in this flower, we've got both the white and the pink showing up. That would be more analogous to codominance. And in both of these, it is the heterozygous individual that is changing a little bit, because neither trait is truly dominant or truly recessive. And there's another better example of codominance that we will see in just a little bit. Another example of this would be Sax Disease. This is an inherited disorder in humans, and what happens is that the brain cells of affected children cannot metabolize certain lipids, which cause the buildup of those lipids in the brain and eventually leads to death. So people with Sax Disease do not live particularly long, unfortunately. But here's the thing: only the homozygous individuals will have the disease. So there's your normal kind of dominant recessive. If we look down a level, right, kind of at a biochemical level of the individual, the heterozygous individuals have half the normal levels of the enzyme activity. Now, half of that normal level is still enough to prevent the buildup. But if we kind of are looking at that level, that's going to be incomplete dominance, right? That's kind of a blending of the two, either not having it all or having it. So you're kind of in the middle there. However, if we actually look at the enzymes themselves, half of the enzymes that produce are dysfunctional and half are functional, which would be an example of codominance, right? Because you're getting both of the traits actually showing up. However, one of them is kind of able to make sure that the disorder doesn't take over. So that's kind of an example and a way to look at kind of the different levels of the dominance. Now, overall, again, kind of on the organismal level, it's just dominant and recessive, because you either have the disease or you don't. But as we kind of look down at different layers, we're able to see a little bit more nuance. So we also can have these kind of multiple things being interacting. We can have multiple alleles. Here's where we have more than 2 allele options for each gene. However, there are only 2 inherited still, because you only have 1 of each chromosome. So you still only have 2 places to have the alleles. However, there might be 3 or 4 or 5 different alleles that you could inherit. A good example of this is blood type. We can also have polygenic inheritance. Here, more than one gene affects the outcome of one character. So, for example, skin color is affected by multiple genes, or height is affected by multiple genes, things like that. So over here, we've got an example of multiple alleles. So for blood type, there are actually 5 different choices that you can have. We're only going to look at the 3 right here. So we've got AB blood, right? So you can either have the A antigen, which is on the outside of your blood cells. You can have the B antigen, You could have both, or you could have none. So there are 3 different alleles that code for those 4 options. You're either IA, IB, or lowercase I. Okay. A and B are co-dominant. So if you have the A allele and the B allele, you will have both of those antigens on the outside of your blood cells. If you have only the A antigen, then you're only going to have the A. If you have only the B, you have the B. If you have lowercase I with either A or B, it is recessive to those. So A shows up over I and B shows up over I. So the only way to have O blood is to have I as your allele. Okay. Over here we've got polygenic traits. So here we've got 6 different genes that code for 3 different genes that code for the skin color. And you can basically, depending upon how many of those alleles turn out to be dominant or recessive, that is going to affect you on a scale. So here we've got 6 of the dominant alleles. Here we've got 6 of the recessive alleles. Okay, so the number of alleles is what is going to determine on a scale, and that's one of the reasons why we have such a wide variety of skin colors, things like height, hair types, all these different types of things. Most of the things that in our body are polygenic, especially our outward characteristics, and so because of that, we can have such a wide variety of them in the population. All right, now there are a couple things that make things even a little bit more complicated. We've got pleiotropy, where one gene does not necessarily affect only one character, but it can affect many others. An example of this would be sickle cell. So those folks that have sickle cell anemia, it doesn't just only affect one thing, it actually affects many others. Epistasis is another one that makes things a little bit more complicated. And basically, it's the inheritance of one allele affects the expression of another. So in the example over here, black is dominant to brown. So you see that we've got more black mice forming than brown mice. However, without a dominant C allele, neither pigment gets deposited in the fur and the mouse becomes white. So the individuals that are homozygous for the lowercase c, they do not have any color, even though they have the dominant, they would have the dominant black allele. They're not able to produce that pigment, or rather, they are able to produce the pigment, but doesn't stay in the fur. Okay, and so that would be an example of epistasis. So, just kind of a summary of these different things. We can have either complete dominance, we can have incomplete dominance, we can have codominance, multiple alleles, pleiotropy, epistasis, and polygenic inheritance. So we have a famous person, Thomas Hunt Morgan, who in the early 1900s, he, along with his grad students, were investigating genetics using fruit flies. The reason why they were using fruit flies, very fast life cycle. They produce a lot of offspring per breeding season, and they only have 4 pairs of chromosomes, so it's very easy to kind of track what's going on. They have 3 pairs of autosomes and 1 pair of sex-determining chromosomes. And they discovered sex-linked genes and linked genes, which are another type of inheritance that can occur. Let's talk just briefly about sex determination. In mammals, we've got the XY system, where if you have 2 X's, you're a female. If you're an X and a Y, then you are a male. However, that is not the only system out there in the animal kingdom. So in some organisms, we have the ZW system. The reason why they're using Z's and W's is because it is the opposite. So in humans, homozygous means female. For chickens, homozygous means male. So we want to have a little bit of distinction there. So that's why they call it the ZW system. We also can have the X naught system. So here, XX equals a female, and X by itself is going to be a male. And then finally, we've got the haplodiploid system. So if you are a diploid, you're a female, and if you're a haploid, then you are a male, and that is used in bees and some other insects as well. So we're going to focus on flies and humans. So genes are located on the sex chromosomes. Many more of them are located on the X chromosome, as we have discussed. And so therefore, most of the time when we talk about sex-linked genes or sex-linked traits, we're talking about things that are located on the X chromosome, because the Y chromosome is so small, it just doesn't have a lot of genes. Because of this, males exhibit a much higher frequency of these X-linked traits than do females. The reason why? Males only have 1 X chromosome, so they don't have any opportunity to kind of hide a recessive trait behind a dominant allele. So some examples of this in humans are colorblindness, hemophilia, and Duchenne muscular dystrophy. So let's take a look at kind of how that's inherited. So this would be an example of colorblindness. So we've got a female that is a normal, total normal person, homozygous dominant, and then we've got a male who has colorblindness. There's no way for that male to pass on the colorblind gene to his sons, because he's always going to pass on a Y chromosome to his sons. However, he is going to pass it on to his daughters, and his daughters are going to become what we call carriers. So they're not actually exhibiting the trait, but they are carrying the gene along, so that's why we call them a carrier. A female who is a carrier can then have children with a normal male and have an opportunity to produce a colorblind male. However, there's not going to be an opportunity to create a colorblind female, because the normal male will always pass on the normal gene for color recognition, and so therefore the females will only be a carrier. And then finally, the males, a male who is colorblind, if he were to have children with a female who was a carrier or would be colorblind, it is possible to have a female that is colorblind. But that requires that you have to have some have a fairly long odds, right? You have to have a carrier female and a colorblind male. Now, we talked about this a little bit in class, all right? This idea of X inactivation. So during embryonic development, so probably somewhere around in the 16 to 32, maybe up to 64 cells, some of the X chromosomes begin to turn off or become inactivated in mammals. The reason why, you don't want to have all those extra proteins being produced, because that could lead to developmental problems. All right. So what happens is that one of the X chromosomes gets inactivated and becomes what is known as a bar body, and this results in the normal expression of the proteins, as well as this phenomenon known as mosaicism. So you can see that in the cat right here, right? The cat has both orange and black fur. The reason why, some of the cells, when they were inactivated, carried the orange color, some of them carried the black color. So you're left with some of the cells expressing the orange color fur, some of them expressing the black color fur. That's kind of known as a turtle shell cat in terms of the coloration. And now some of you might be wondering, okay, what happens in meiosis when you've got this bar body? Are half the gametes going to be unable to function? That's not going to be the case. In the germline cells, they're going to reactivate the bar body so that all the gametes are going to have an active X chromosome. And this is only going to occur in females. It's not going to occur in males. In humans, this can occur with females in terms of the sweat glands. So if females are heterozygous for the trait for sweat glands, you can have females that have kind of patches of skin where they don't actually have sweat glands, because the cells are in that patch have that gene shut down because it was on the bar body. So kind of interesting. All right, linked genes. This is a little complicated. All right, genes located near one another on the same chromosome. So these are on the same chromosome, right? Which means that it's going to be more difficult for them to be inherited independently of one another, right? Because they're on the same chromosome. The same chromosome is going to get passed back and forth. Now, linked genes can be separated or can be inherited separately by crossing over events, right? So crossing over occurs, those linked genes get shuffled around, and now they can be inherited separately. How close they are on the chromosome is going to impact on how often this crossing over occurs that results in a recombinant chromosome. So if the genes are closer to one another, they're more likely to stay together. Does that make sense, right? Because they're not going to be, it's not going to be as likely that a crossing over event occurs just in between them to get them to swap. The farther apart they are, the more likely they are to be to cross over, because there's more likely that a chiasmata can form in between them during meiosis. So what we can do is we can use it to create a linkage map, which shows us the relative positions of genes on chromosomes. Oops. One map unit, which is a artificial unit that is created simply for this. It doesn't correspond to anything except for just a percent chance of crossing over. And these result in what are known as recombinant offspring. So the DNA has recombined. Let's kind of take a look at this. What we do is we take, and this is Hunt again, he came up with this. He took a wild type fly and crossed it with a wild type means that it's like kind of the normal trait. And so he took the wild type and he crossed it with a double mutant. So these are two different traits, body color and wings. And so he crossed them together. And what that produces was the F1, which is wild type, but it's heterozygous for both traits. Okay, so it's heterozygous for both traits. He then takes that and crosses it with another double mutant, right? And what we get is the range of different offspring that we would expect. So we get some of them that are wild type with the normal wings, some of them that are double mutant, like we'd expect. We also get some that are gray with vestigial wings, and some of them that are black with normal wings. So notice that we have different types than what we started with the parents, right? We've got a recombining of the chromosomes. Now, if the genes are located on different chromosomes, we would expect to have an equal ratio of all of them, right? Because they're going to be recombining they're going to be all independently inherited, right? Independent assortment, so that we should have an equal number of each of them. If the genes are located on the same chromosome and they're always inherited together, so if they're like really close together, then we would expect to have all either gray normal or black vestigial, right? Because they're going to move with one another. So those are the offspring that we should always get. However, our actual results resulted in a higher number of the gray normal and black vestigial than the gray vestigial black normal. However, we still got some of those, okay? So let's take a look at kind of what that means. So we ended up with, these are the chromosomes, right? So the egg cells from the female either were the normal, right? Either had were from the original grandparents, right? So we're originally either from the original P generation, okay? Right? So this is the P generation chromosome. Here's the other P generation chromosome. Or a crossing over event has occurred, and so now we create these recombinant chromosomes, okay? With either B plus VG or B VG plus, okay? We then combine that with the sperm, which we know, because it's a double mutant, is going to be BVG, all right? And so we have 4 different combinations that can occur. 2 of them are the parental offspring. These are the ones that we would expect to have, right, normally. 2 of them are the recombinant offspring with the recombinant chromosomes located inside, all right? What we do is we take the total number of recombinant offspring, we divide it by the total number of offspring, multiply that by 100 to give us our percentage, and we get 17%, which means that the genes B and VG must be 17 map units apart. We can then use that and compare it with another gene, and we can create our linkage map. So here's another gene, CN, right? Its recombinant frequency with B is 9%. Recombinant frequency with VG is 9.5%. So we know that it's somewhere in the middle between the 2 of them. It can't be over here, right? Because if it was over here on this side, right, over on the right of VG, then we would expect there to be a higher recombination frequency between B and CN than between VG and CN, okay? Does that make sense, right? So we've kind of got this, we've got a way to be able to relatively lay out maps. And this is what they were doing before the Human Genome Project, when they actually, sequenced all the DNA. They were doing these linkage maps to figure out where the different genes were located on the chromosomes. Pretty cool stuff, but it is a little complicated. All right, some other forms of inheritance. Remember, mitochondria and chloroplasts have their own DNA. This is inherited maternally, right? In humans, at least, because egg cells contain more mitochondria than sperm cells. Egg cells are much, much larger than sperm cells, so they are the ones that are going to be passing on the mitochondria. This is used a lot to determine human ancestry. You know how people have, we talk about how humans came out of Africa. The reason why we know that is we follow back the mitochondrial DNA. And you can see how humans have kind of spread out around the world, and you can see how we have come to inherit different parts of the world using mitochondrial DNA and seeing where different groups split off evolutionarily. We also have this idea of epigenetics, which has kind of been used a lot as kind of this weird catch-all term. But what it really means is that is a trait that is inherited that is not tied directly to what your DNA says or what your alleles are. And there are kind of 2 different ways that this can occur, although these could also be the exact same thing. This is a really brand new way of looking at inheritance. So we've got a genomic imprinting. So a trait is only expressed when inherited from a specific parent. So maybe it's inherited from the mother, but not the father. So that offspring shows the trait, but then the other one inherits it from the father, but not the mother, and doesn't express the trait. Now, the reason why this could come into play is this idea of methylation. So methylation is methyl groups getting added onto the DNA. And when the methyl groups get added onto the DNA, it causes the DNA to kind of pack in tightly. So then your enzymes that are necessary to read the DNA and then produce the proteins from it can't get at it. So that would not allow the trait to be expressed, right? Even if you have the DNA, if you don't have access to the genes, you can't actually produce the proteins that are gonna allow you to express the trait, right? So that would be that is one of the things that people believe, or scientists believe, are going to kind of cause this idea of genomic imprinting and this whole epigenetics idea. Finally, a little note on nature versus nurture, right? The environment has a large impact on how genes are expressed. Why do you think the government tells mothers not to drink during pregnancy? Right? Alcohol can affect the development of the child because it's going to have an impact on how genes are expressed. Same thing with taking drugs, smoking. All those types of things have an impact during pregnancy. There's lots of people talk about their parents or their mother eating lots of random foods, and then they really like those foods. There could be some sort of impact there. Also, hydrangea flower color, and this is true for other plants as well, but this is kind of a classic example. It's controlled by pH. So as the pH of the soil becomes more acidic, the flowers actually become a deeper blue, right? As it becomes more neutral, it becomes more of a purple-pink color. So kind of cool stuff there. And that's all I've got for you today. I know this stuff is a little complicated, but you can definitely handle it. Thanks for watching, and I'll see you next time.